Welcome to the deep dive. Today, we're going to be going pretty deep into the world of spectroscopy.
Yeah.
It's something that, maybe a lot of folks don't think about on a daily basis,
Right.
but it's pretty vital in making sure that all the medicines we rely on are safe and effective.
Absolutely.
Think of it kind of like a detective working behind the scenes in the pharmaceutical world,
Exactly.
making sure everything is up to par.
Making sure that that pill that you take actually has what it says it has in it.
Right.
And that it's pure and it's going to work the way it's supposed to.
Absolutely.
Yeah.
And to really get into this, we've got a pretty interesting stack of sources. We've
Sure.
got everything from really in-depth scientific articles that get into the nuts and bolts of each technique,
Right.
all the way to some pretty serious regulatory documents that spell out what's required from a quality standpoint.
Absolutely.
So it's going to be a pretty comprehensive view.
Yeah.
Our mission today is really to unpack these techniques,
Right.
understand how they're applied across the entire lifespan of a drug,
Yes.
from its initial development to its large-scale manufacturing.
Yeah. From start to finish.
Yeah,
How do these things ensure the quality of the final product that ends up, at your pharmacy or in your medicine cabinet?
absolutely.
Yeah.
So first off, what are these techniques that we keep mentioning?
So spectroscopy, in a nutshell, is the study of how matter interacts with electromagnetic radiation.
Okay.
Now, that might sound a little bit complicated, but think of it this way.
Yeah,
By analyzing the patterns of absorption or emission of light,
right.
we can get a ton of information about the composition and structure of a substance.
Right.
And we're going to focus on a few key methods that are widely used in the pharmaceutical industry.
Got it.
And they all work together to make sure that the medicine you take is good.
Let's start with UV-Vis spectroscopy.
Okay.
I think this one, a lot of people, maybe they've heard the name,
Sure.
but maybe they're not exactly sure what it is.
So UV-Vis spectroscopy, or ultraviolet visible spectroscopy, is based on a pretty simple principle.
Okay.
It looks at how molecules absorb light in the ultraviolet and the visible parts of the electromagnetic spectrum.
Right.
The amount and the pattern of that absorption is unique to different compounds,
Right.
and it's related to the electronic structure.
Okay.
So really what UV-Vis lets us do is get a really reliable head count of the molecules that we're interested in.
So you can use it to identify what's there
Absolutely.
and how much is there.
Exactly.
Kind of like using shining a light through a colored solution and seeing how much of that light gets through.
Absolutely.
And based on that, you can get a feel for the concentration.
You got it.
Okay, so UV-Vis, it's kind of like a molecular fingerprint based on how it absorbs light,
Precise.
and it's used to identify things and quantify, right?
Yes.
You mentioned the regulatory aspects too.
Yeah, so this ties in directly with making sure that, pharmaceutical manufacturers are following the rules.
Okay.
Things like 21 CFR 211, which is a regulation that dictates how drugs should be manufactured.
Right.
And that ultimately affects the quality of the medicine that you receive.
All right.
Yeah.
So that's UV-Vis.
Yes.
What about infrared or IR spectroscopy?
Okay.
I heard about that.
So IR spectroscopy looks at a different part of the electromagnetic spectrum.
Okay.
This one tells us about how molecules vibrate.
Vibrate.
Yeah.
Oh, right.
So when a molecule absorbs infrared light of a specific wavelength, it causes its bonds, its chemical bonds, to start vibrating,
Huh.
right? And those different functional groups, different parts of a molecule, they vibrate at characteristic frequencies.
Okay.
So, for example, a carbonyl group or a hydroxyl group, these are different parts of a molecule.
Right. Yeah.
They're going to vibrate at different frequencies.
Got it.
So think of it like IR spectroscopy is giving each molecule a unique vibrational signature. And that allows us to really pinpoint what functional groups are there.
So you're looking at what wavelengths of light are absorbed.
Exactly.
And from that, you can infer the functional groups that are present.
You got it.
Okay.
Yeah.
So it's like identifying the ingredients in a recipe based on, if you knew what each ingredient tasted like, you could kind of tell by the overall flavor
Exactly.
what's in there.
Okay.
So IR is good for identifying materials and figuring out the structure based on these functional groups.
Absolutely. It's like identifying the specific building blocks within a molecule,
Okay.
which is really important to make sure that the drug you take has the right structure.
Got it.
Yeah.
So we've got UV-Vis for identifying compounds, quantifying them.
Right.
We've got IR for the functional groups.
Yes.
Now there's this thing called Raman spectroscopy, which sounds a little bit different.
It is a little different. So Raman spectroscopy, it actually looks at the scattering of light. So not absorption, but scattering.
Right.
And specifically, it looks at inelastic scattering.
Inelastic.
Yeah.
Okay.
So when light interacts with a molecule, most of it's going to scatter off with the same energy that it had when it went in.
Right.
But a small fraction of it will be scattered inelastically, meaning it loses or gains a little bit of energy.
Okay.
So its wavelength changes slightly.
Got it.
And we call that change in wavelength the Raman shift.
Okay.
And that gives us information about the molecule's vibrational modes.
Okay.
Kind of similar to what we were talking about with IR.
Got it.
But it's a slightly different approach.
So instead of looking at what light is absorbed, you're looking at how the light changes when it scatters off the molecule.
Exactly.
Okay.
Yeah, it's like shining a light on something and seeing how it wiggles the light back.
Right.
And those wiggles tell us something about the molecule structure.
Got it.
Yeah.
Okay, so you mentioned inelastic scattering.
Yeah.
So the light kind of, it loses or gains a little bit of energy.
When it interacts with the molecule.
Yeah.
And that's kind of the basis of the technique.
That's how we get the information.
Okay.
Yes.
And you were saying that this technique has some advantages over the others.
It does in
Okay.
some cases.
So one of the cool things about Raman is that it can often be used for non-destructive analysis,
Okay.
meaning you don't have to destroy your sample to analyze it.
Right.
So you can imagine if you had a medicine, a sealed medicine, you could actually analyze it through the packaging
Wow.
without ever having to open it.
That's pretty impressive.
Yeah,
Okay, so non-destructive.
non-destructive.
What else?
Another advantage is that water doesn't scatter Raman light very strongly.
Okay.
So this makes it really useful for analyzing aqueous solutions, right?
Right.
Things that are dissolved in water or biological samples,
Got it.
which are very common in the pharmaceutical world.
Right.
Yeah.
And didn't you mention something about it being used in like manufacturing?
Yes. So Raman actually has a really great potential for in-process monitoring.
Okay.
So what that means is you can actually use it while you're making a drug.
Wow.
So for example, Raman spectroscopy can be used to monitor blend uniformity.
Okay.
So in continuous manufacturing, you're making a tablet, right? You're mixing up all these powders, the active ingredient, with other things.
Right.
And you want to make sure that it's all mixed evenly.
Absolutely.
So Raman can be used to check that in real time.
Okay.
And if something goes wrong, if it's not homogeneous, it can actually trigger an alarm.
Wow.
And you can adjust the process right away.
So it's like a feedback loop
Exactly.
for quality control.
Real-time quality control.
Right there in the manufacturing line.
Exactly.
That's incredible.
Yeah, it's a really powerful technique.
Okay, so UV-Vis, IR, Raman. Now we got to talk about NMR.
Okay, yeah.
Nuclear magnetic resonance. That one, I always got to admit, that one always sounded pretty complicated.
It's definitely the most intricate of the techniques that we've talked about.
Okay.
But it gives you just incredible detail about the structure of molecules.
Okay.
So the way it works is you place a sample in a really strong magnetic field,
Okay.
and then you expose it to radio frequency radiation.
Okay.
Now, certain atomic nuclei, like hydrogen-1 and carbon-13, they can absorb this radiation,
Right.
and then they emit it back out at specific frequencies.
Okay.
And those frequencies are sensitive to the magnetic environment of that nucleus.
Okay.
So basically, you're listening to those nuclei as they send out these signals.
So it's like you're tuning into a radio station, but the radio station is a specific atom in the molecule.
Exactly.
And it's telling you something about its environment.
Exactly. It's like a little radio transmitter inside the molecule.
Okay.
Giving us information about what's around it.
Got it.
Yeah.
And from that, you can kind of get a feel for the connectivity. Like,
Yeah.
how are all these atoms connected?
You can figure out the bonds between the atoms.
Okay.
You can figure out the three-dimensional shape of the molecule.
Wow.
You get a lot of information from NMR.
So that one is definitely the most detailed.
Yes, absolutely.
Okay.
Yeah.
You were saying that's particularly good for organic compounds?
It is. It's really the gold standard for identifying and characterizing organic compounds.
Got it.
And in fact, our sources talk about how it's used in drug development,
Okay.
right? So when they're doing organic process research and development,
Right.
they're actually using NMR to figure out, are they making the right molecule?
Got it.
And that helps ensure that the drugs are safe and effective.
So it's like it's used across the board in a lot of...
It's a very versatile technique.
Yeah.
Yeah.
All right, so we've got these 4 techniques.
Yes.
They all provide different information about the molecule.
Right.
Now, how are these things used in quality control,
Okay.
specifically in the pharmaceutical world?
So one of the primary roles of these spectroscopic techniques is compound identification.
Okay.
So at every stage of the drug-making process, you need to confirm what you have.
Right.
Right, so you start with your raw materials,
Right.
you have your intermediates as you're making the drug,
Right.
and then, of course, the final drug product.
Got it.
So you need to make sure that you have the right thing at each step.
Right.
So we do that by comparing the spectrum of an unknown sample to a reference standard.
Okay.
So the reference standard is basically a known pure sample of the thing that you're trying to make.
Okay.
And so if they match, you're good.
Right.
You know you have what you're supposed to have.
And that's super important, for making sure that,
It's absolutely critical.
yeah, the drug that ends up on the shelf is actually what it's supposed to be.
Yeah, you don't want any mix-ups, right?
Right.
And again, the regulations are really clear about this. Things like 21 CFR 330, which talks about the identity, strength, quality, and purity of drug substances.
Right.
So you have to be able to prove that you have what you say you have.
Got it.
And spectroscopy is the way to do that.
So it's like having a whole library of fingerprints,
Right.
but instead of fingerprints, they're molecular fingerprints that you can use to compare against.
Exactly. It's like a molecular database.
Okay, so that's compound identification.
Yes.
What about purity?
Purity is absolutely critical as well.
Right.
And spectroscopic techniques can also be used to detect and quantify impurities.
Okay.
So if you're looking at a spectrum and you see some unexpected peaks or deviations from the pure standard, that's a sign that something else is there,
Right.
that something might be a contaminant.
Okay.
And that's important because impurities could affect the efficacy of the drug.
Right.
They can also have toxic effects.
Right.
So you really want to make sure that your drug substance is as pure as possible. Yeah. And our sources actually talk about, how much effort goes into this.
Okay.
Right. So there's a handbook on isolation and characterization of impurities,
Right.
which goes into detail about all the different ways that you can try to isolate and identify these impurities.
Right.
Right. And there are even organizations like the PQRI, the Product Quality Research Institute,
Right.
which specifically focuses on mutagenic impurities.
Okay.
So, these are impurities that could potentially cause mutations in your DNA.
Wow.
So there's a lot of work that goes into making sure that the drugs are free from those kinds of impurities.
So it's not just a matter of like, oh, is it pure or not? It's like, are there any specific impurities that are going to be particularly problematic?
Exactly. Yeah. And you need sensitive methods to detect those.
Got it.
Yeah.
All right.
So we've talked about compound identification, purity.
Right.
Now, what about the actual formulation itself? So the final pill or the liquid that you would take, how does spectroscopy play a role there?
Right, because it's not just about the active ingredient.
Right.
It's about how it's all put together
Absolutely.
into the final product.
So we can use spectroscopy to characterize the formulation.
Okay.
So, for example, we can use IR and Raman to look at the solid state of a drug.
Solid state.
Yeah, so things like polymorphism.
Polymorphism. Now that's where things get a little bit
Polymorphism is basically the ability of a solid to exist in different crystal structures.
Okay.
So the same molecule can pack together in different ways.
Got it.
And that can affect things like solubility
Okay.
and bioavailability, how well the drug is absorbed by your body.
Right.
And we actually have a whole book on polymorphism in our sources.
Okay.
That goes into a lot more detail about that.
Got it.
Yeah. So IR and Raman can tell us about those different crystal structures.
Okay.
And then NMR, it can even be used to study reaction conversion during formulation.
Right.
So how much of the starting material has actually been converted into the final product?
Got it.
And it can also be used to look at the impact of different additives
Okay.
on the formulation.
Got it.
So things like preservatives or stabilizers that are added to the drug
Uh-huh.
to help it last longer.
Right. All the inactive ingredients.
Exactly.
Okay.
Yeah.
So it's really about understanding not just the active ingredient itself,
Right.
but how the whole formulation behaves.
Absolutely. How does it dissolve? How stable is it?
Right.
All these things are important.
Okay, so we've talked a lot about, what each of these techniques can do.
Yes,
Now, every tool has its limitations.
of course.
So let's talk about some of those.
Okay.
Like, where does each of these techniques fall short?
So yeah, each one has its advantages and disadvantages.
Right.
So starting with UV-Vis, it's very sensitive,
Okay.
and it's really good for quantitative analysis,
Right.
especially for compounds that absorb UV or visible light. We call those chromophores.
Right. So those are compounds that are good at absorbing those specific wavelengths of light.
Exactly.
Okay.
However, it can sometimes lack specificity,
Okay.
meaning that different compounds might have similar absorption spectra.
Right.
So it can tell you that something's there, but it can't always tell you exactly what it is.
So it's like if you're trying to identify a fruit based on its color.
Right.
There's a lot of red fruits.
Exactly.
Yeah. Right.
So it narrows it down, but it doesn't give you the definitive answer.
Right.
Yeah.
Okay.
So that's one limitation of UV-Vis.
Got it.
Now, IR spectroscopy, that gives you a lot more structural information
Okay.
because it's sensitive to those functional groups we talked about.
Right.
So it's better at identifying compounds.
Okay.
However, sometimes you need to do some sample preparation to get a good spectrum.
Okay.
And certain materials, like water, can interfere with the measurement.
Right.
Yeah.
Okay.
So that's something to keep in mind with IR.
What about Raman?
So Raman, one of the big advantages there, we talked about it, is that it's non-destructive.
Right.
You don't have to mess up your sample.
Right. You don't have to, dissolve it in something or crush it up or anything like that.
Exactly. You can analyze it as is,
Okay.
and you can analyze a variety of sample types,
Oh, right.
including aqueous solutions and even through packaging.
Right.
So that's a big plus.
Yeah.
However, the signals can be kind of weak sometimes.
Oh, okay.
So you need a more specialized instrument to get a good signal,
Right.
and those instruments can be a little more expensive.
Okay.
So that's something to consider with Raman.
And finally, NMR, which, we said is kind of the most powerful in terms of structural information.
Yeah, NMR can tell you so much about the structure.
Right.
How the atoms are connected, the 3D arrangement, everything.
Right.
But it is less sensitive
Okay.
than the other techniques.
Right.
So you need more sample.
Okay.
And it takes longer to get the data.
Right. So it's not really ideal if you're dealing with very low concentrations or if you need to analyze a lot of samples very quickly.
Right, if you're doing high throughput screening or something like that.
Exactly.
Okay.
And the instruments themselves, the NMR instruments, they're quite expensive.
Okay.
So that's another factor to consider.
So it sounds like these techniques, they all have, strengths and weaknesses,
They do.
and they're kind of complementary in a way.
They're all part of the toolkit.
Yeah.
And you choose the right tool depending on the job.
Right, like if you need to quantify something very quickly, UV-V might be the way to go.
Exactly.
But if you need to really get into the weeds of the structure, NMR might be the answer.
Exactly. Or if you have a sample that you don't want to destroy, you might go with Raman.
Right. Depends on the situation.
Exactly.
Okay, now, to really bring this to life, do we have any case studies?
We can infer some potential case studies based on the information we've read.
Okay.
So let's imagine a scenario.
Right. Let's say a pharmaceutical company receives a batch of an active pharmaceutical ingredient.
Right.
And they use IR spectroscopy to analyze it.
Okay.
And they find that it's missing a key functional group that should be there.
Okay.
That's a huge red flag.
Right.
It could mean that it's counterfeit or that it's degraded somehow,
Right.
and so they don't use it.
Right.
And that prevents potentially dangerous product from reaching patients.
Right.
So that's a case where IR spectroscopy really helped protect public health.
Absolutely.
Yeah.
Okay. How about an example with Raman in a manufacturing setting?
Okay, so let's say you're making tablets in a continuous manufacturing process.
Right.
And you're using Raman spectroscopy to monitor the drug content in the powder blend,
Okay.
right, as it's being mixed.
Right.
And let's say the Raman spectrum starts to show some deviations,
Okay.
meaning that the blend is not homogeneous.
Right. Some parts of the mixture have too much drug and some parts don't have enough.
Exactly. So the system can actually trigger an alarm,
Right.
tell the operators to stop and adjust the process.
Okay.
And that prevents a batch of tablets from being produced
Right.
that don't meet the specifications.
So again, it's kind of like a real-time quality check
Exactly.
that prevents problems.
Right. It ensures that every tablet has the right amount of drug in it.
Okay.
Yeah.
What about NMR?
So NMR is often used to deal with impurities.
Right.
So let's say in drug development,
Okay.
they're making a new drug
Right.
and they detect an impurity
Okay.
and they want to figure out what it is.
Right.
So they use NMR to analyze the structure
Okay.
and they're able to figure out where this impurity came from in the synthesis.
Okay.
And they can modify the process to prevent it from forming.
Got it.
So that's a really important application of NMR.
So it's not just identifying the impurity.
Right.
It's like using that information to change the process,
To improve the process.
to make a better product.
Exactly. Got it.
Yeah.
And finally, a lot of folks are familiar with the idea of a pill or a liquid having a certain concentration
Right.
of the active ingredient.
Right.
How does UVV come into play there?
So UV-Vis is often used for just that,
Okay.
to determine the concentration of a drug.
Okay.
So, if you have a bottle of cough syrup, it's going to say on the label it has this much, active ingredient per milliliter.
Right.
So UV-Vis is used to verify that,
Right.
to make sure that it actually has the right concentration.
So that's like a day-to-day quality control type of test.
Absolutely.
Okay.
And it's required by good manufacturing practices, or GMP,
Right.
which is a set of regulations that ensures the quality of pharmaceuticals.
Right. So all these techniques, we see them in kind of every aspect of drug development and manufacturing.
They're involved at every stage.
Yeah.
From the very beginning, when they're first discovering a new drug, all the way to the end.
Right.
Mhm.
When it's sitting on the shelf at the pharmacy.
Right.
Yeah.
So let's talk about that a little bit.
Yeah.
Like, where do we see it early on in development?
So in early drug development, spectroscopy is really important for characterizing new chemical entities.
Sure.
So when they first make a new molecule that they think might have some therapeutic potential,
Right.
they need to figure out what its properties are.
Right.
How soluble is it? How stable is it?
Right.
How does it interact with other things?
Right.
So spectroscopy can,